Chapter 12, the Cardiovascular System. So just to warn you, it's at this point that the lectures are going to start getting much longer just because the chapters are longer, and so we'll end up covering much more material. Here are the primary objectives for this chapter, so just feel free to pause the video to read them, and I'll wait just a second here. Section 12.1, the heart ensures continual 24/7 nutrient delivery. So the cardiovascular system is the organ system which transports blood, which contains nutrients, wastes, and dissolved gases, to and from the tissues. Now I know in your book it sometimes calls the system the circulatory system, but we're going to call it the cardiovascular system throughout this lecture, just because the term circulatory system is kind of inaccurate, and cardiovascular is a much more scientific word, so that's the word we're going to use. And the cardiovascular system works with the respiratory system, which brings oxygen to the blood and then removes carbon dioxide from it, and we'll cover that in the next lecture. So the cardiovascular system consists of three basic component parts: the heart, the blood vessels, and the blood. And the cardiovascular system in vertebrates is called a closed circulatory system, and here we'll use the term circulatory because it's fine to use here. And this means that the blood remains within the confines of the blood vessels and heart and never directly bathes the tissues of the body. So all vertebrates actually have a closed circulatory system, as do worms and cephalopods like octopus and squid. But an example of an open circulatory system would be what insects have. And in an open circulatory system, the interstitial fluid and the blood are not separated, and they actually bathe the cells, or rather the cells are bathed in this fluid. And we're not going to get any more into that because it's really beyond the scope of this class. Here's a basic schematic representation of the cardiovascular system. So just note a couple things here. So over in the lungs is where we take in oxygen from the air we breathe and then remove carbon dioxide from the body and put it back in the air. And then at the top and bottom, where the cardiovascular system is delivering blood to the body tissues, we see that we're removing carbon dioxide and then bringing in oxygen to those tissues. And we'll talk about the chambers and some of the larger vessels here in a second. The heart has 3 layers, 4 chambers, and is a 2-cycle organ. So the heart is surrounded by a membrane called the pericardium, which secretes a lubricating fluid which surrounds the heart, and this allows the heart to beat without causing damage to itself. The heart is contained within a cavity called the mediastinum, which is part of the thoracic cavity, and this contains the heart, major blood vessels, and the lymphatic vessels of the lymphatic system. The heart has 3 layers. There's the epicardium, or outer layer, the myocardium, or middle layer, which is a muscular layer, and this is what contracts and causes heartbeats. And then finally there's the endocardium, or inner layer inside the heart. The heart also has 4 chambers. There's the right atrium, which receives blood from the body's major vein, the vena cava, and then brings the blood into the right ventricle. The right ventricle is a portion of the heart which pumps blood into the lungs through the pulmonary artery. The left atrium receives blood from the lungs by way of the pulmonary veins and then brings it to the left ventricle. Now the left ventricle is the most powerful ventricle of the two, and this is because it has to pump blood throughout the body, and the blood leaves this chamber of the heart through the body's major artery, the aorta. Here's a diagram of the three layers of the heart. So we have the pericardium here in blue, and the layer of the heart nearest the pericardium is the epicardium. The layer in the middle is the myocardium, or the muscular layer, and then in purple is the endocardium. Here's a diagram of the structure of the human heart, and I'm just going to point out where the chambers are. So on the upper right portion of the heart, or our upper left, is the right atrium, and then below that is the right ventricle. And then up at the upper left of the heart, or our upper right, is the left atrium, and below that is the left ventricle. And of course, at the top of the heart in red is the aorta, which connects to the left ventricle. And then in blue is the vena cava, and this is over towards the right of the heart, our left. And the vena cava actually connects to the right atrium from both the top and bottom, so it's coming at it from both the top and bottom. And then finally, over on the top of the heart, on our right, is the left pulmonary artery. And we're going to cover how blood flows through the heart and then through the rest of the cardiovascular system here in these next few slides. Each heart chamber contains a valve. So each of the four chambers in our heart contains a valve which opens to allow blood into that chamber and then closes when the ventricles contract. And what valves essentially do is to prevent backflow of blood into the chamber it came from, and this makes the cardiovascular system much more efficient when we prevent backflow. So there are two types of valves. There are atrioventricular valves, which have this name because they're between the atria and ventricles. There's the tricuspid valve, which is between the right atrium and right ventricle, and this valve has three cusps, which are basically flaps that make up the valve, and we'll look at those in a second. And then there's the bicuspid valve, which is called something else in the book. I think they just use the two names interchangeably. We'll talk about that in a second. And this is the valve between the left atrium and left ventricle, and this has two cusps. And then there are the semilunar or atrial valves. Both of these have three cusps, and these are just the valves at the top of each atrium. There's the aortic valve, which leads into the heart, and then there's the pulmonary valve, which leads into the pulmonary artery. And these are shaped like three flexible bowls and are anchored to the blood vessel walls. Valves are all composed of dense, irregular connective tissue and are held in place by cordae tendineae, which literally translates to cords of tendons. These are also called heartstrings, and they anchor the cusps of the valves to the papillary muscles, which we'll talk about more in the following slides. Here's a diagram of the heart valves. In this diagram, we're looking over the top of the heart. At your bottom right, you can clearly see the bicuspid valve and the tricuspid valve. And obviously the bicuspid valve has two cusps, and the tricuspid valve has three. And then over at your bottom left, you can see the cusps of the semilunar valves, and both of these have three. And just note that when The tricuspid valve and the bicuspid valve are open to let blood into those atrium. The semilunar valves are both closed. But when the heart beats and we move blood into the lungs and into the rest of the cardiovascular system, the semilunar valves are open, whereas the atrioventricular valves are closed. Heart valves can make a variety of sounds. They can flutter, hum, rasp, or tap, and all of these sounds mean something specific. So if the opposing surfaces of a valve fit poorly, or if the cusps of the valve fit poorly, the blood can spill past and cause the valve to flutter. And yeah, I know I typed past, it's pass. This creates a murmur, which is an audible change in heart sound, contrary to the normal heart sound. And this can possibly lead to valve prolapse, which is to say the backflow of blood. And then you can have mitral valve prolapse, and mitral valve is another name for the bicuspid valve. And this occurs when the mitral or bicuspid valve fits together poorly. This is usually runs in families, and it's more common in women than men. And this just also leads to the backflow of blood out of the bicuspid valve. The pulmonary and aortic valves produce a tapping noise as they feel and knock against one another, and this of course is a normal heart sound. This is actually heard as the second portion of the heartbeat. Continuing from the last slide, auscultation means to listen to heart sounds. It actually means to listen to any type of organ sounds in the body, but for the purpose of this chapter, we're just going to say that we're listening to the sounds that the heart makes. So the sounds that the heart normally makes are called S1 and S2, or lub and dub, because that's the sound that is made. So S1 occurs as a loud resonating sound caused by the blood pressure against the atrioventricular valves. So this sound occurs when the bicuspid and tricuspid valves close, and this pulls the chordae tendineae and the entire supporting framework of the muscle, or rather it pulls on it and causes it to contract. The S2 sound is caused when the ventricles relax and the blood in the pulmonary artery and the aorta flows back towards the ventricles. And this isn't backflow into the ventricles, it's just backflow against the valves. So it's the atrial valves which catch the backflow, and this is the sound of them snapping, or rather the cusps snapping against each other. If the ventricles are slightly out of sequence, such that one closes before the other one does, the S2 sound may occur as a stutter or as a split, and a constant split may indicate hypertrophy of one ventricle. And hypertrophy is when an organ or part of an organ increases in size or volume, and usually when this occurs in the heart, it might point towards some other problem that a person may be having in other parts of their cardiovascular system. In addition to making the sounds we just talked about, the heart can also murmur. Now, your book doesn't do the best job actually telling you what a heart murmur is, but it's basically either an unusual or an extra sound made by the heart, described as a whooshing, blowing, or rasping noise, and this occurs when blood passes the valves in a turbulent flow, which changes the sound or the number of sounds. And this may signal a serious valve problem, but a lot of times it's also harmless, or can be harmless. So not all murmurs are cause for alarm. Sometimes children develop heart murmurs as they grow, simply because the cardiac muscle grows faster than the valves grow. And pregnant women may develop a murmur as the result of a dramatic increase in blood volume. So when a woman's pregnant with a fetus, the fetus is going to obviously need its own blood supply, so that's the reason why there's this increase in blood volume. The heart is called a two-cycle organ because blood flows through the heart twice. So now we're going to trace the flow of blood through the heart. So first, blood enters the right atrium from the superior and inferior vena cava, and the superior vena cava comes at the right atrium from the top, and the inferior vena cava obviously comes at the atrium from the bottom. Once blood enters the right atrium, it drops through the tricuspid atrioventricular valve into the right ventricle. The right ventricle then pumps the blood into or out of the right side of the heart via the pulmonary valve, and the blood then enters the pulmonary trunk, which is the vein that leads to the lungs, and then blood enters the lungs, where it is oxygenated or takes up oxygen from the air we breathe in. So then blood returns to the heart from the lungs, entering the left atrium. Once it's in the atrium, the blood drops through the bicuspid valve into the left ventricle. The left ventricle then pumps the blood out of the heart via the aortic valve of the aorta, and then the blood circulates throughout the body and eventually ends up back at the heart again. So when a chamber of the heart contracts, this is called systole, and when a chamber of the heart relaxes, it is called distole. And also remember that when ventricles contract, the atria relax because they've already contracted and pushed the blood into the ventricle, so there's really no need for them to be contracted as the ventricles contract and push the blood wherever it needs to go. So here's a diagram of the cardiac cycle, which pretty much is just a picture representation of what I just said. So you can see the blood entering the right atrium from the vena cava. It then enters the right ventricle, which then pumps the blood towards or into the lungs via the pulmonary trunk. And then after the blood's been oxygenated in the lungs, it enters the left atrium, and then it enters the left ventricle, and then the left ventricle contracts and pushes, or rather forces the blood throughout the body. And now we're just going to look at what I started talking about in the last part of that slide. So in part A here, we're in distole, which means both the ventricles and the atrium are both relaxed. Atria. Atria is the plural of atria, or atrium, Then we enter the atrial systole phase, and this is just when the atria are contracted and push the blood into either ventricle. And then after the atria have contracted, the ventricles enter systole and contract, and like I said, the right ventricle sends the blood to the lungs, and the left ventricle sends blood throughout the body. Now we're going to talk about the typical blood pressure changes that occur in the heart. So I don't know if I did such a good describing what these words mean in the last slide or two, but systole means the state of contraction, and distole means a state of relaxation. So systolic pressure is the measure of the force produced by the left ventricle contracting, and this increases with age. whereas diastolic pressure is the force the blood exerts on the walls of the cardiovascular system while the heart is completely relaxed. And this cannot be zero. If it is, you are dead and have been drained of blood. So just having blood in the heart produces some sort of pressure, obviously. And normal blood pressure is measured or defined as 120/80 millimeters of mercury, and a high blood pressure is defined as 140/90 millimeters of mercury or higher. And we need to maintain proper blood pressure because it is essential for maintaining homeostasis throughout a whole organism. The rate at which your heart beats is actually under two types of controls, and we'll go into more detail about these in the following slides. So we have intrinsic controls, and these controls establish the usual day in and day out pace of our heartbeats. And then we have the extrinsic controls, which modulate the baseline heart rate to meet the body's immediate blood demands. So because the intrinsic controls control heart rate on a day-to-day basis, they need to create synchronized contraction. Cardiac muscles undergo rhythmic contractions without receiving nerve impulses, which is a very good thing because we wouldn't really want to think or send a conscious thought through our brain each time the heart needed to beat. So when the intracellular calcium concentration reaches threshold in the heart muscle cell, the cell itself spontaneously contracts. And when two or more cardiac muscle cells touch one another, they can begin to beat in unison, following the pace of the fastest cell. And this is something that would be seen if we removed two individual heart cells from the heart and put them together. So the actual part of the heart that controls or serves as the intrinsic control is the SA or sinoatrial node. And this is the node which is called the heart's pacemaker. And it's actually a group of cells in the upper wall of the right atrium near the entrance of the coronary sinus. Or this is basically just a blood vessel which brings blood to the actual heart tissue. And this group of cells has the fastic intrinsic beat. So when the SA node actually initiates heartbeat, the signal to contract passes in wave-like fashion from cell to cell through the right and then the left atrium, causing both of these atria to contract. And a healthy heart rate is usually around 60 to 100 beats per minute, and there might be some variance in this depending on if we're relaxing or if we're exercising or not. Continuing with intrinsic controls, so there's another node in the heart called the AV or atrioventricular node, and this is a group of cells at the base of the left atrium near the ventricle. And these cells serve as a relay station that delays the contraction impulse before sending it on. And this node serves as a relay station between the SA node and the AV or atrioventricular bundle. And the delay it creates allows the atria to complete their contraction before the ventricles are stimulated to begin contracting, because we don't want all of these contracting at the same time. After the delay, the impulse sent from the SA node passes through a series of conductive tissues before reaching the cells of the ventricles. So in order to obtain a simultaneous contraction of both ventricles, the ventricles themselves require a conduction system from the contraction impulse. So this contraction impulse, and we're going to trace the flow of it, travels from the SA node to the AV node, then the AV bundle, and then the right bundle branches to what are called the Purkinje fibers. And the Purkinje fibers are conductive myofibers or muscle fibers that end at clusters of ventricle cells. And these fibers ensure that the impulse gets to these cells simultaneously to allow for the synchronous contraction such that the entire ventricle contracts. When intrinsic control of the heartbeat no longer works or functions properly, the heart muscle cells lose their ability to contract in a synchronized fashion. And one example of this is a condition called ventricular fibrillation, in which the heart has, or rather, the electrical control over the timing or rhythm of the heartbeat is lost. And in this condition, the ventricular cardiac cells can no longer beat in a coordinated fashion, and this means that they can no longer create a contraction forceful enough to either push blood into the lungs if it occurs in the right ventricle, or push blood into the aorta or the vessels connected to it if it's the left ventricle that's affected. So then this causes the heart to stop beating, which actually is a condition called cardiac arrest. So this just means that the heart stops. And since the heart has stopped, this can lead to sudden death. And just keep in mind that ventricular fibrillation is different from a heart attack, as a heart attack results from the blockage of a coronary artery. A coronary artery is an artery which feeds blood to the heart tissue itself, and we'll look at those in the next slide. And this cuts off oxygen supply to the tissue, which leads to the death of that tissue. So apparently your book doesn't do a really good job of telling you what coronary arteries are, so I just copied this slide and put it here so we can go over what they are. So if you look at these really thin red arteries attached directly to the heart, these are the coronary arteries, and their job is to feed blood into the actual cells of the heart so they don't die of lack of oxygen. And just note that the reason we have these arteries instead of just sending the blood directly to the heart when it is pumped or flows into the heart is because the blood can't actually cross the endocardium layer, or rather it can't cross into the endocardium layer in the heart, so it's much more efficient just to send blood to the heart through these arteries, especially designed for that purpose. But anyway, that's what coronary arteries are. Moving on to extrinsic heart rate controls and how they can override intrinsic controls. So the extrinsic heart rate controls are the controls used when the body needs more blood than the resting or normal heart rate delivers. And this is actually a cardiac control center in the medulla oblongata portion of the brain. And this part of the brain can override the intrinsic heartbeat, leading to an increase or decrease in heart rate as necessary. So this would happen when we're producing norepinephrine or epinephrine in response to some sort of threat in the fight or flight response. And this would, of course, lead to increased heart rate. And then we have Stirling's law, which is when the ventricles are stretched by increased blood volume, and then they recoil with matching force. The increased blood flow to the heart is what causes the heart to respond with more forceful pumping, as what happens when we exercise, because we need a higher rate of blood flow to account for all the oxygen we're going through as we exercise, and energy also, in addition to oxygen. Now we're going to talk about electrocardiograms, which are graphs which record the electrical activity of the heart. And sometimes we replace the C with a K, and this is how the English spell electrocardiogram. So an electrocardiogram, or ECG as we call it in the United States, records the pattern of electrical signals generated by the cardiac muscles as they contract and relax. And these graphs have a defined series of peaks and valleys within them. So a P wave shows when the sinoatrial node fires, which causes the atrial cells to depolarize. And this is why I covered the nervous system earlier, so we would know what this means. And this causes atrial systole, or atrial contraction. Then the QRX complex is a part of the graph created by the simultaneous depolarization of many ventricular cells. And this is where the ECG is momentarily flat as the ventricles are in systole or a contraction state. In the T wave of the graph, the ventricles are relaxed and the cells repolarize. And then they return to a state of relaxation or dystole. The PR interval is the part of the graph which shows atrial depolarization to ventricular depolarization. The contraction impulse is transmitted from the SA node, travels through the atria to the AV node, and then finally to the conduction system of the heart. If the PR interval happens over a period of longer than 0.2 seconds, this may indicate damage to the conduction system of the AV node. And then the QT interval is the total time of ventricular contraction and relaxation. If this is a long interval, it may indicate congenital heart defects, conduction problems, coronary ischemia, I'm sorry I probably just butchered that word, or a previous heart attack. So here's an electrocardiogram where we've labeled the peaks and valleys that we see in these graphs. So the PR interval is just when we go from point P to point R. The QRS complex involves point Q, R, and S. And then the QT interval just goes from the valley at Q to the peak of T. And then it clearly shows where the PR interval is and the QT interval is, along with the normal periods of time where the heart either remains in atrial systole, ventricular systole, or in diastole. Section 12.2, blood transport involves miles of sophisticated plumbing. So now we're going to begin talking about the blood vessels and not the heart so much. So there are three types of blood vessels. There are the arteries, which take blood from the heart to the tissues, and these always are taking blood away from the heart to another destination. The capillaries are extremely thin-walled diffusion vessels within the specific tissues of the body, which actually is where oxygen exchange occurs. And then there are the veins, which take blood back from the tissues to the heart. And veins are always bringing blood back from the tissues to the heart, and that's kind of the best way to remember the difference between arteries and veins, because arteries take blood from the heart and veins take blood to it. So the walls of both arteries and veins, but not capillaries, have three layers. There's the endothelium, or the innermost layer. The middle layer is a muscular layer made up of smooth muscle and elastic tissue. And then finally, the outer layer is made up of connective tissue. Arteries have thicker, smooth muscle and connective tissue layers than veins, as they experience higher pressures, as their taking blood away from the heart, they're more subject to the actual pressures created by the ventricles actually contraction. And another difference between arteries and veins is that veins have valves sort of like those in the heart, which prevent blood backflow. And just remember that valves always prevent blood backflow. So here's a diagram which shows the structures of arteries, veins, and capillaries. And just over here at your right is an artery, and it shows the three individual layers of that blood vessel. And just remember that the lumen is the actual hollow part of the blood vessel through which blood flows. And over at your right, you can clearly see the valve in this vein, and usually valves are only in the larger veins. And then finally we have the capillary, which is basically just 2 cells thick because we have a layer of endothelium and a layer of basement membrane cells. Once again, arteries carry blood from the heart. So the arteries are on the output or ventricular side of the heart, and that's the location they would need to be in order to accommodate carrying blood away from the heart. And those arteries closest to the heart have the largest diameters and the thickest walls out of all the arteries because they experience higher fluid pressure here. And they're experiencing this as a result just of the pressure produced by the ventricles. Farther from the heart, the arteries become smaller in diameter and the wall thickness decreases. And this causes an increase in friction, which slows blood flow, just because the smaller the vessel, the larger the surface to volume ratio. And eventually arteries become arterioles as they become smaller, and these obviously branch from larger arteries and have a very similar structure. Blood flow is obviously slower through arterioles because they have a larger total cross-sectional area than the arteries do, even though they're smaller in diameter. And this means they have smaller lumens, which expose the blood to more surface area, which creates friction and further slows blood flow. Capillaries are the blood vessels through which oxygen exchange occurs, and they are the smallest blood vessels. And if you wrote down that capillaries are 2 cell layers thick, you better make that 1, because they're only 1 cell layer thick. And their lumens are just big enough for 1 erythrocyte or red blood cell to cross through. And like I said, due to this 1 layer, they permit the vital exchange of gases and also nutrients and wastes across the blood vessel wall. And capillaries reach nearly every cell because every cell is going to need oxygen. And a capillary bed is an interwoven mat of capillaries which threads through a tissue. The blood flow in the capillary beds is regulated by precapillary sphincters, which are little ring-shaped muscles which close or open parts of the capillary bed, depending on the oxygen and nutrient demands of the tissue. So just here, remember that not every cell needs a constant or near constant supply of oxygen. They can go without oxygen for a little while, and we don't need to be or have a constant flow of oxygen to every cell all the time. Veins bring blood back to the heart. So first we're going to start with venules, which are basically just smaller veins, because they're the veins which connect to the capillary beds, because now we're moving from smaller vessels to larger ones. So these are small veins that drain blood from capillaries to the larger veins. Now the veins themselves, as do the venules, bring blood from the tissues back to the heart once again. Veins have thinner walls than arteries as they experience little pressure, and this is just because blood flow has just slowed to this extent due to friction in the arteries. Despite the low blood pressure in the veins, blood continues to flow towards the heart, and this flow continues because fluids flow easily from smaller vessels to a larger one, as there's a reduction in friction as we go from smaller to larger. And since the veins have valves in them, or at least the large ones do, this helps to prevent any backflow of blood and blood pooling. And blood pooling is just when we have a bunch of blood pooling together, as the term would suggest. And these valves counteract the effects of gravity. Also, the contraction of the skeletal muscles prevents backflow, as when these contractions occur, they squeeze the veins and push the blood back towards the heart. And edema is a condition which results in abnormal swelling in tissues due to blood pooling, and exercise is often prescribed, and it's pretty understandable why exercise and movement would cure this problem or solve this problem, is because it increases just the rate of blood flow throughout the body. So here's a diagram that shows the exchange flow in capillary beds, and this just explains how gas and exchange, and also waste and nutrient exchange occurs. So when we're wanting to take in gases and nutrients, we need a blood pressure that is higher than the interstitial fluid and osmotic pressure leaving the cells, such that we can have flow from higher concentration to lower concentration. And then when we're wanting to remove waste and carbon dioxide from the cells, we need a blood pressure which is lower than the interstitial fluid and osmotic pressure, such that we remove these wastes. Section 12.3. Different cardiovascular pathways have specific purposes. So, as the heart is described as a two-cycle organ, blood can take one of two pathways from the heart, and these pathways are the pulmonary circuit and the systemic circuit. So in the pulmonary circuit, we transport blood to and from the lungs, such that we exchange carbon dioxide in the blood for oxygen from the environment. And then in the systemic circuit, we transport blood to and from the tissues. And in this circuit, we bring oxygen and nutrients to the tissues and remove carbon dioxide and other wastes from these tissues. Once again, in the pulmonary circuit, carbon dioxide is exchanged for oxygen in the lungs. So let's go into more detail about the pulmonary circuit. It extends from the right side of the heart to the capillary beds in the veins, and then to the left atrium where it ends. So in this circuit, the deoxygenated but carbon dioxide rich blood enters the right atrium, which is then sent to the right ventricle. And like we've said before, the right ventricle pumps the blood into the respiratory membranes of the lungs, where it can pick up oxygen and release carbon dioxide back into the atmosphere. The now oxygenated or oxygen-rich but carbon dioxide depleted blood then returns to the left atrium, and this completes the circuit. In contrast to the pulmonary circuit, the systemic circuit delivers oxygen to the tissues and returns carbon dioxide to the environment. And this circuit extends from the left side of the heart to the capillary beds of the tissues, and then back to the right atrium. Rich blood or oxygenated blood enters the left atrium. Blood is sent to the left ventricle, and the left ventricle pumps blood through the aortic arch of the body. After passing through the capillaries, the blood in the veins returns to the superior and inferior vena cava. And at this point, the blood is deoxygenated, but carbon dioxide rich, and it enters the right atrium. And then we enter the pulmonary circuit again. Here's a diagram which shows the routes of circulation in both the pulmonary and systemic circuits. So in the pulmonary circuit, it begins at the right atrium, blood flows to the lungs, and it officially ends before it enters the left atrium. And then the systemic circuit begins when blood enters the right atrium, is pumped throughout the body by way of the left ventricle, and then it ends right before it enters the right atrium. So now we're going to talk about coronary arteries again. I should have looked at this slide before I pulled up that other one. Anyway, coronary arteries are the first branches from the aortic arch which deliver oxygen-rich blood to the cardiac muscle. And this is because the endocardium is not a diffusion membrane. The cardiac tissue must obtain oxygen through a capillary bed just like all the other tissues. However, these arteries are narrow and prone to clogging, and when these arteries are blocked, this is what can lead to a heart attack. Now we're going to talk about portal systems. Blood flows from arteries to capillaries to veins, and then the veins break up into another set of capillary beds, and this is what a portal system is. They allow the blood to flow into a second organ before pushing it back to the heart. So one of the major portals in the body is the hepatic portal, which drains blood from the small intestine to the liver, and then the blood passes through another capillary bed. This is what allows the hepatocytes or liver cells to remove detrimental ions, compounds, and other wastes that are picked up in the digestive tract. The cleansed blood then collects in the hepatic vein and drains to the inferior vena cava. So just kind of remember what a portal system is, and keep in mind what the hepatic portal leads into and what's done to the blood by the liver there. Section 12.4. Cardiovascular disorders have life-threatening consequences. So now we're going to go over some of the most common cardiovascular diseases, and these include hypertension, atherosclerosis, heart attack, heart failure, embolism, stroke, and varicose veins, which is the least severe of these. And we'll talk about these in more detail in the following slides. There are several risk factors for developing cardiovascular disease, or CVD, and these risk factors can either be genetic or environmental. Genetic risk factors include a family history of CVD, and one such example of this is that a family history of heart attack prior to age 55 likely indicates a genetic predisposition to heart disease. Gender is also another genetic factor. CVD is more common in men than women. And then there's also ethnic background, which would likely determine your odds of developing a CVD and also determines what sort of CVD you may develop. Other risk factors are environmental. There's smoking, which is kind of just all around bad for you, overeating, and not exercising. And some studies indicate that only 40 minutes a day of low-impact exercise reduces the chance of heart disease by 1/3. So these are some pretty telling studies. High blood pressure stresses the entire body, so the more technical term for high blood pressure is hypertension. And this is diagnosed when the systolic blood pressure is above 140 millimeters of mercury, or the diastolic pressure is above 90 millimeters of mercury. A high diastolic number indicates a decline in the elasticity of blood vessels, and that increases the chance that the force of systolic contraction will exceed the capacity of the circulatory system. High blood pressure is harmful to many organs, but stroke is our greatest concern here, as your chances of having a stroke increase. And a stroke is when the blood flow to the brain is interrupted or reduced, and these are essentially the same as a heart attack, they just occur in the brain. Two genes are involved in the conversion and activation of the protein angiotensinogen, which is the protein that causes blood vessels to constrict. If this pathway of angiotensinogen is hyperactive, constricted vessels will result, and this will increase your blood pressure. So we do not want more angiotensinogen than what we need to function properly. Artery damage is a major cause of mortality and disability. Atherosclerosis, which literally translates to hardened vessels, is a disease of the blood vessels. And this hardening of the blood vessels occurs when plaques or fatty deposits of cholesterol accumulate inside the vessel walls. And this causes some blockage of the lumen, which reduces blood flow to certain parts of the body. Serious complications can arise if these plaques cause a clot to form within the blood vessel. And a thrombus is a clot that is still attached to the vessel wall. However, these can sometimes be large or loose, and these large loose clots are called embolism, and they float throughout the bloodstream. A big danger with embolisms is that these clots can lodge in a smaller vessel, completely blocking blood flow to that organ and causing tissue death. And when this happens in the coronary arteries of the heart, we have a heart attack. And when it happens in the arteries and blood vessels of the brain, we have a stroke. An aneurysm is what occurs when a vessel wall balloons under pressure, and this is actually caused by the destruction of the muscle layer of the blood vessel, such that the blood vessel just keeps expanding. And these aneurysms actually form a weak spot that can burst with the pressure generated by your heartbeat. And when aneurysms burst, they can cause death. However, they can sometimes be repaired before they burst by replacing the balloon area with plastic tubing. And over here you can see an aneurysm that I'm surprised hasn't caused death already, just because look how big it is. But you can kind of get an idea of how serious these can be. So stroke can be caused either by an embolism or an aneurysm in the brain, both of which stop blood flow to the brain. And this results in the starvation of the tissues fed by the affected artery of oxygen and nutrients. The initial symptoms of stroke include sudden difficulty speaking, blindness in one eye, and numbness and or weakness, usually only on one side of the body. And this is related to the fact that we just have two hemispheres in the brain which control either side of the body. Stroke can also cause aphasia or loss of speech, loss of fine motor control, paralysis, and even death. there's a new emphasis on the quick treatment of strokes, which has reduced disability resulting from this. And obviously, the reason this results is because we can't really regrow brain tissue. So now whoever's had a stroke could possibly become mentally handicapped. And obviously, strokes will cause widespread brain damage, like I said. So here's just a chart of some of the steps to take if you suspect someone has had a stroke. So first, you look at their face to see if one side of the face is droopy compared to the other side. You ask that person to raise both arms, and if one arm drifts downward while the other remains up, that's a sign. You need to check and see if the person has difficulties speaking or if their speech is slurred or strange. And then obviously, if both of these three are happening, you need to get them medical attention pretty quickly so we don't experience any mentally damaging effects. Here's more about heart attacks or myocardial infarctions, MIs. And like I said before, a heart attack leads to the death of a portion of the heart muscle tissue due to a lack of oxygen caused by diminished blood flow to that tissue. And heart attack is the number one cause of, or rather causes 1 in 5 deaths in the U.S. And of course, when the cardiac muscle tissue dies, the nervous or electrical impulse can no longer be passed through the heart muscle tissue, and this greatly reduces cardiac output of the heart. So a heart attack is usually caused when a plaque or thrombus becomes lodged in a coronary artery such that we formed an embolism, and this causes a great reduction in blood flow to that tissue. So before a heart attack occurs, and while the plaque is forming, the blood flow is beginning to diminish at this point, and this can cause the heart tissue to act like a cramped muscle because of the reduced amount of oxygen getting to that tissue. And this causes a crushing feeling in the chest, pain that radiates throughout the chest and left arm, or numbness in the left arm, and this cramping of the cardiac muscle is called angina. So angina may arise when the heart is working too hard or is under more stress than usual. And there are actually 2 types of angina. There are stable and unstable angina. Stable angina can be treated simply by reducing activity and or possibly quitting smoking, whereas unstable angina isn't quite so curable, and it appears with no apparent stimulus, and this is usually the type of angina that serves as an early warning sign of impending heart attack, and this would cause probably all of those symptoms we saw in the last slide. And this would mean that we need immediate medical attention if these symptoms are occurring. But unstable angina may be controlled with nitrate drugs, such as nitroglycerin, yes, that's the chemical that's in dynamite, and isosorbide. And these drugs relax the smooth muscle walls of the coronary arteries such that the blood pressure in these arteries decreases and blood can flow more smoothly and thus pass over or through the obstructive plaque. So now we're going to talk about 2 surgical strategies to counteract the effects of plaque formation in coronary arteries. So the first of these is balloon angioplasty, in which a catheter with a deflated balloon at the end is threaded into the artery. The balloon is then inflated such that it expands the lumen, and this pushes and flattens the plaques against the vessel wall, such that we now have a more cleared lumen. And then very similar to a balloon angioplasty is a stent, which is essentially a rounded meshwork of wires, metal pieces, which supports the arterial walls, and this permanently opens the blood vessel and improves blood flow. And these stents can be coated with medicine, which block plaque buildup. And these medicines leach from the stent, and they provide a constant dose of whatever drug we're using. And then finally there's bypass surgery, which may be required if the coronary artery is either physically damaged or the plaque buildup is so severe that the other two options won't work. So bypass surgery is much more invasive than angioplasty or just putting in a stent, because it is an open heart surgery. And in order to perform the surgery, the breastbone or sternum is broken to reach the heart, and then the heart is periodically stopped so that the surgeon can perform the delicate suturing. So in order to perform this surgery, first a vein, or rather the femoral part of the femoral vein, is removed, and this serves as the bypass vessel. Surgeons then suture or sew, which is what suture means, a small length of this vein around the blockage in the coronary artery, and this works something like a highway detour. This allows blood to bypass the congestion and return to normal circulation and go around the area which is blocked. And each of these detours counts as one bypass, and that's how we end up naming these surgeries. So a triple bypass would involve 3 of these detours. And usually this surgery is highly effective, but recovery is much slower just because of all of the suturing and breaking of the breastbone and cutting into the patient. So here's a diagram from your book which shows how these surgeries work to improve blood flow in the artery. So let's start with part B, which shows balloon angioplasty. And in this surgery, they'll shove a catheter or tube into the artery. And in the case of balloon angioplasty, they inflate the balloon, and this pushes the plaques up against the walls of the arterial lumen, such that we improve blood flow. And then in part C shows a stent in a coronary artery, and as you can see, this is just a cylindric metal meshwork that they use a catheter to shove into that artery, and this permanently opens the artery and improves blood flow. And over here in part A is a bypass surgery where they've taken this vein from the femoral vein, and they've sutured the vein to the aorta of the heart, and also sutured it to the coronary artery, such that we've bypassed this plaque that you can see. Congestive heart failure is a condition in which the heart has become too weak to push blood through the cardiovascular system. When the left ventricle is weakened and fails to move blood, this causes fluid to back up and leak into the lungs, which leads to a condition called pulmonary edema. And if you'll recall, edema is a condition where we have pooling of blood or other fluids into an area that we don't want it to pool into. So as the blood flow slows because of the weakness of the left ventricle, fluid tends to build up elsewhere, such as in the thoracic cavity. And this causes fluid to press up against the heart, such that beating becomes much more difficult. Congestive heart failure is a gradual disease, and the body actually attempts to compensate for this weakness of the heart. The heart tissue itself expands and enlarges the volume of each ventricle. The enlarged volume requires more muscle mass to push the blood, such that we increase muscle mass, and this actually causes a further expansion in the heart. Tachycardia also results, which is an abnormally rapid heartbeat, and pushes even more blood throughout the body until the heart can no longer maintain the rapid pulse. And symptoms of congestive heart failure include fatigue, difficulty breathing, and tachycardia. Fluid builds up in the lungs and can drown the respiratory membranes, which of course leads to death because we can no longer exchange gas in those membranes. Now we're going to sort of take a step back and talk about a couple non-lethal conditions. One of these conditions is varicose veins, which are distensions of the venous walls near valves. And these are caused when blood moves into the veins and pulls against the valves of the veins. And if the walls of the vein are weak, either due to disease or genetics, this causes the vessel to expand. They expand even more as each heartbeat brings more blood into the distended area. The vein then pops out of the musculature and becomes visible as a bluish wrinkled cord directly beneath the skin. And the only treatment for this is to surgically remove that vein. And they're very unsightly, but are not lethal. Spider veins are essentially the same thing as varicose veins, except they occur in the venules, or small veins. And these are visible through the skin as pale purple or blue tracings, and they usually occur in small to large patches on the face or thighs. And unlike varicose veins, we can treat these by injecting the venule with sterile saline solution, which displaces the pooled blood. And these really can't cause too much damage. They're just mostly cosmetic, and they usually happen in older individuals. Sometimes hearts that appear to be healthy can also fail, and here are 3 conditions which could cause heart failure in a seemingly healthy individual. There's hypertrophic cardiomyopathy, or HCM, which is simply an enlarged heart. Your book says that this is a syndrome of heart myocardium outgrowth, which can usually be detected with ultrasound, though usually when individuals experience heart failure due to this condition, they don't know that their heart is enlarged until it's too late. Another condition is atherosclerosis, which, like we said before, is the hardening of the arteries such that these arteries can be subject to embolism or aneurysm during athletic activity. Another condition is arrhythmia, which is essentially just erratic heart rhythm. But this can also cause athletic heart failure because extreme exertion or exercise sometimes triggers this condition, which can be fatal. But for the most part, experts say that exercise helps the heart and overall function of the cardiovascular system because it strengthens the cardiac muscles and the vasculature by improving the overall efficiency of circulation and respiration. So exercise is still good for you, but in extreme or rare instances, it can be lethal. Section 12.5, blood consists of plasma and formed elements. Now we're going to leave the heart and the rest of the cardiovascular system and just talk about blood for the rest of the chapter. Blood consists of two things: plasma, which is the liquid component, and the formed elements, which are the solid components of blood, and these are mostly cells. Blood is critical to maintaining homeostasis for these reasons. It regulates the internal environment of the body by allowing diffusion of ions and other materials into the interstitial fluid. And if I haven't said it before, interstitial fluid is simply the fluid between cells. Blood forms clots to prevent loss of blood after injury. It transports heat between the core of the body and the skin, serves as a mode of transport for the hormones, nutrients, and gases dissolved in the plasma, and the formed elements in blood deliver oxygen and patrol the body to destroy invading pathogens. So here's a centrifuge tube of spun blood, and if you don't know, a centrifuge is simply a machine that we stick these tubes or vials in that then spins them around such that we've separated the components of blood. But anyway, you can see up at the top of it we have plasma, which consists or makes up 55% of blood, and as it's the lightest part of the blood, it's situated at the top. And here, a little bit heavier, we have the white blood cells and platelets of the blood suspended in this small white band. And then below that we have the red blood cells in red here at the bottom because they're the most dense. And all of these cells are what make up the formed elements in blood. And as you can see or tell by this little white band, there's relatively few white blood cells and platelets in the blood, though there's enough apparently. Plasma makes up 46 to 63% of total blood volume. Plasma is 92% water, 7% dissolved proteins, and 1% electrolytes or dissolved ions, nutrients, and wastes. The proteins in blood help maintain the blood's osmotic pressure, and the osmotic pressure ensures water will remain inside the vessels instead of diffusing into the surrounding tissues. And this is because proteins are simply too big to pass through the capillary walls, so they stay in the blood. And the main protein found in the blood is albumin, which is actually the protein responsible for the white pigment of an egg yolk. But anyway, it's in human blood also, and if the albumin level drops, the osmotic pressure of the blood shifts, and this will force water from the blood vessels into the tissues, which causes edema, which is again pulling of fluids into an area that they shouldn't be pulled into. Albumin binds with several drugs, or rather binds to several drugs, including penicillin, and it's responsible for helping transport drugs similar to penicillin. The electrolytes in plasma are mostly sodium and potassium. Nutrients in plasma include carbohydrates and amino acids, and the wastes are mostly urea, carbon dioxide, and lactic acid, which is produced, of course, as a byproduct of exercise. And urea is just a nitrogenous waste, and we'll talk about that more in an upcoming chapter. So the formed elements of blood are cells and cell fragments, all of which originate from the red bone marrow. And this red bone marrow is located within the apices of long bones, such as in those in the hip and sternum in adults. Of course, in children, red bone marrow is located in nearly all bones. And these cells within the bone marrow are stem cells, which are capable, in this case, of differentiating into erythrocytes or red blood cells, thrombocytes or platelets, or leukocytes, white blood cells. And we're going to call them by their scientific names. Differentiation of these stem cells occurs under the direction of hormones and colony stimulating factors. The erythrocytes account for 99.9% of the formed elements in blood, whereas the leukocytes and thrombocytes account for the remaining 0.1%. The development of the stem cells in the bone marrow into mature blood cells is called hematopoiesis. And here to your right is a diagram of the various blood cells that the stem cells differentiate into. So over here in the diagram at your far left, it shows a red blood cell or erythrocyte, and as indicated by the diagram, red blood cells eject their nucleus at some point in time during development, just so they can carry more hemoglobin molecules and thus carry more oxygen. Then there are the platelets, which are actually released from these much larger cells called megakaryocytes, and platelets are essentially just cell fragments. And then all the rest of these cells are immune cells or leukocytes, of which there are two different types, which we'll talk about in the next slide. But anyway, we have eosinophils, basophils, neutrophils, and these are granular leukocytes. And then we have monocytes, T lymphocytes or T cells, and finally B lymphocytes or B cells, which are agranular leukocytes. Leukocytes are blood cells specialized for the defense of the body against foreign invaders or pathogens, and thus are a critical component in the immune system. There are approximately 5,000 to 11,000 leukocytes per cubic centimeter of blood, compared to 4 to 6 million erythrocytes per cubic millimeter of blood. So there's much fewer leukocytes than there are erythrocytes. And there are 5 types of leukocytes. Like I said with the diagram, there are 3 types of granular cells: the neutrophils, eosinophils, and basophils. And I'll tell you why they're named this in a second. And then there are 2 types of agranular cells: lymphocytes and monocytes. Leukocytes are impossible to identify with the naked eye, so we must use a stain to identify them. And when these cells are stained, they either develop granules or they don't. And these granules are dark spots or particles in the cell that appear in the cytoplasm, and we can view all of this under a microscope. And the granulocytes get their names from what happens when they are placed in Wright stain, which is the stain used for identifying leukocytes. Neutrophil granules only stain with the neutral stain, or the stain with a neutral pH, and thus they are neutral loving or neutrophile. Eosinophil granules only stain with a stain called eosine, which is a bright orange or pink color, and thus they're called eosinophils because they love that stain. And then finally, basophil granules take on the color of the basic stain used to identify them, and their granules are nearly black. And like it says, the pH of the stain is pH 11, so it's very basic. When agranular leukocytes are stained, they don't pick up the stain because they have no granules. And there are two types of agranular cells. There are lymphocytes, which are small round cells with little visible cytoplasm. They're mostly nucleus. And they come in two varieties. There are the B cells, which produce antibodies, and we'll talk more about this when we cover immunity. And then we have T cells, which act directly against the pathogen, and they ingest the pathogen by way of endocytosis. And then finally we have the second type, monocytes, which are the largest of all the white blood cells, including granular leukocytes. And these contain quite a bit of cytoplasm, surrounding a large kidney-shaped nucleus. So here's just some pictures of these blood cells after they've been stained. So over here at your far right, we have an eosinophil, which you can see has picked up the reddish pink stain eosine, and then we have a basophil, which has picked up the basic stain such that its granules are rather dark. And then we have the neutrophil in the middle. And then we have the lymphocyte, and as you can see, it's a small cell which has little cytoplasm and is mostly nucleus. And finally, we have the monocyte, which is a rather large cell over at right. Here's a chart from your book which shows the number, characteristics, and functions of the various formed elements in blood. I realize that the heading on this slide doesn't exactly make sense with what's being shown on this chart, but it's the heading from your book, which is shown before the book shows the chart. So I just put it up here so you could find this chart easier. But anyway, I've kind of doctored the chart so that we have the red blood cells and the platelets together here. Here's the rest of the chart, which shows the leukocytes, which we just went over. And you can just use this chart to quickly go through the formed elements in blood. And I think it's probably going to be a pretty good study aid for the test. Now we're going to talk about red blood cells, or erythrocytes, which are the cells responsible for carrying blood throughout the body. Now erythro translates to red, and cyte means cell, so these are red cells. Each of these cells can carry approximately 200 million molecules of hemoglobin protein. Now each and every single one of these hemoglobin protein molecules contains 4 molecules of a pigment called heme, which is just a chemical group within that hemoglobin. Now each of these heme groups is attached to an ion of iron, such that each hemoglobin molecule has 4 atoms of iron at its center, and it's actually this ion of iron that allows hemoglobin to bind to oxygen, because iron has a positive charge, whereas oxygen has a negative charge. Essentially, blood is red for the same reason that rust is red, because when iron binds oxygen, this creates iron oxide, which is the chemical name for rust. Hemoglobin is capable of responding to the varying oxygen pressures or oxygen demands of the body tissues and blood. So when we're in an environment where oxygen content is very high, iron tends to pick up oxygen and hold on to it, because the body tissues don't really need oxygen so much when there's so much of it available. Compare this to an environment where oxygen is scarce, and in this environment, hemoglobin needs to release the oxygen so it can be available for the body tissues so they don't die of oxygen starvation. Hemoglobin does its job of carrying oxygen throughout the body so well that it is the only respiratory protein found in vertebrates. And obviously there are other oxygen-carrying proteins in invertebrates, but we won't talk about them here. Hemoglobin is also capable of responding to changes in pH and temperature. Such an environment where the pH is low but the temperature is high would be found, or is rather created, in active muscles when we exercise. And in these conditions, the iron or hemoglobin needs to drop the oxygen so it can be made readily available for the body tissues, because we're going through more oxygen when we exercise. So like I said earlier, erythrocytes lose their nucleus as they mature, and this is so that there's more room for hemoglobin. However, this means that the cell can now no longer repair itself or direct cellular activities, and thus these cells don't survive very long in the cardiovascular system. They suffer a lot of physical damage from continuously passing through the capillaries and blood vessels. So these cells only really survive for 120 days within the cardiovascular system before they are so damaged that they need to be removed from the system. And the organs responsible for erythrocyte removal are the spleen and liver. And these organs break down the erythrocytes and recycle their constituent minerals and proteins, or rather the materials which make up the erythrocyte. And we actually break down about 200 million erythrocytes per second. We also produce about 2 million erythrocytes per second. So we're continuously breaking down and replenishing our supply of red blood cells, or erythrocytes. Here's a picture from your book which shows how tight of a squeeze getting through a capillary is for a red blood cell. So it kind of explains why they get damaged so easily. And here you can see the capillary and the red blood cells just getting squeezed through it. And yeah, that's obviously why they get damaged so easily, because of their continual, or rather repeated, passing through the capillaries. The production and maturation of erythrocytes is called erythropoiesis, and this process is regulated by hormones and our environmental need for oxygen. So when blood oxygen levels drop, the kidneys are stimulated to produce a hormone called erythropoietin, or EPO, which is the hormone that stimulates erythrocyte production. So in general, more erythrocytes means that we have a greater ability to carry oxygen, or we have a higher oxygen carrying capacity. And athletes sometimes take advantage of this fact in one of two ways. Sometimes athletes train at higher altitudes, as the lower oxygen levels usually allow for the production of more erythrocytes, such that the athletes gain a higher oxygen carrying capacity. And this is one fair way of increasing erythrocyte production. However, some athletes use commercially produced EPO to increase erythrocyte production, and this is called blood doping, and it's banned in most sports. However, its competitive advantage is also unproven, because if you increase your amount of erythrocytes too much, it can make your blood so thick that your cardiovascular system has a difficult time pumping it throughout your body. So if you'll recall in chapter 4, where we talked about a structure or layer on the plasma membrane called the glycocalyx, which is made up of glycoproteins and glycolipids. So within this glycocalyx on red blood cells, we have protein markers, or antigens, which are responsible for giving us a specific blood type. So people with the A-type marker or antigen are said to have A-type blood, and people with the B-type marker are said to have B-type blood. And whatever a marker we express is genetically inherited, and these A and B traits are said to be codominant, and this means that some people may have A and B type blood because they express both types of markers. There are also many individuals which express neither the A nor the B type marker, and these people are said to have O type blood. However, they do have, or rather they produce, something called H substance, which is actually modified in people with A and B type blood such that it forms the A or B type marker or antigen. So in people with O type blood, they simply don't modify this H substance, so they don't have the A or B marker. And these blood type markers are really only important when we're donating or receiving blood. And this is because people with type A blood produce a chemical called an anti-B agglutination, or rather anti-B agglutinin, which causes blood type B cells to clump such that they're useless in an individual with A-type blood. And the opposite happens in people with B-type blood when they receive A-type blood, because the anti-A agglutinin causes the A-type blood cells to clump. And people with O-type blood carry both the anti-A and the anti-B agglutinins such that they can accept type A or type B blood. However, people with type A or type B or type AB can accept O-type blood. And if this blood type is not matched before a blood transfusion, obviously the blood cells will be clumped when introduced in the blood, and this makes the transfusion completely pointless and useless. Here's a chart which shows what type of blood individuals can receive based on their own blood type. So over here on the left we have the blood type of the person that's going to receive blood, and over here to the right we have the types of blood that can be safely introduced into that individual. So if you have A type blood, you can accept type A or type O blood, because you're not going to respond to the O type blood because it doesn't carry any markers that would alarm the body. And basically the same thing with B. They can accept type B or type O blood, and individuals with AB type blood can accept both types A and B, and also type AB. And an individual with O type blood can only accept other O type blood because they're going to have that reaction that makes the blood cells clump when we introduce types A or B markers into that individual. Here's a chart which shows the various antigens produced by each blood type, and also it shows the agglutinin produced by individuals of each blood type. So over here on the left we have type A blood, where the A antigen is produced and expressed on the cell membrane, and in these individuals we produce anti-B agglutinin. However, when these cells are exposed to anti-A agglutinin, they clump, as seen here, and this is how we tell this is A-type blood, because the cells clump when they're exposed to anti-A agglutinin. And over here we have B-type blood, which expresses the B-type antigen, and also individuals with this blood type produce anti-A agglutinin. So when we expose this B-type blood to the anti-B agglutinin, the cells clump, and that's how we call it, or rather that's how we tell it's B-type blood, because it clumps with the anti-B but not with the anti-A. And over here we have type AB, which expresses both the A and B antigens and produces neither of the agglutinins. And we can tell that this is AB type blood because the cells clump both when they're exposed to the anti-A and the anti-B. And then here we have type O, which doesn't express either of the antigens, but it expresses both the anti-A and the anti-B agglutinin, such that when we expose it to both anti-A and then anti-B, it doesn't clump with either of them, and that's how we tell it's O-type blood. Blood type is genetically based, but it does vary. Well, it doesn't vary among races, but it varies within races. So here's the blood types in the U.S., and here's the breakdown of blood type among American Caucasians. 46% have type O, 38.8% have type A, 11.1% have type B, and 3.9% have type AB. Now here's the breakdown for African Americans. 49% have type O, 27% have type A, 20% have type B, and 4% have type AB. And then finally we have blood type among Native Americans. 79% have type O, so we have quite a few more individuals with type O blood. 16% have type A, 4% have type B, and 1% have type AB. So here you can see that no matter the race, O is generally the most common, and AB is generally the rarest. And like I said, blood type is genetically based, but it's not really racially based because it varies so much within races. So now we're going to talk about what's responsible for giving us a positive or negative blood type. So your blood cells, or rather your erythrocytes, produce another protein, or rather they can produce another protein called Rh factor, and it's called Rh factor because it was initially discovered in rhesus monkeys, so it's just an abbreviation for that. But anyway, with this type of blood antigen, you either express it or you don't. So if it's present, you're positive, and if it's absent, you're Rh negative. However, people who are Rh negative do not ordinarily have anti-Rh agglutinin. However, once these individuals are exposed to Rh positive blood for whatever reason, their immune system then starts to produce anti-Rh antibodies. And usually this doesn't cause many problems unless an Rh positive man has a child with an Rh negative woman, and this can lead to a hemolytic disease of newborn babies. This doesn't affect the first child the woman has because her blood hasn't yet come in contact with the infant's blood. However, the blood mixes when the infant's born during the course of the birthing process, and now the mother produces this anti-Rh agglutinin. And this is where complications can arise during the second pregnancy. And this is when the infant will get this disease. However, Rh negative mothers can be prevented from producing the anti-Rh agglutinin by being inoculated with anti-Rh positive antibodies immediately after the first birth. And this causes the Rh positive blood to clump, or rather the Rh positive blood of the infant to clump, so that it is then removed from the mother's blood system so that her immune system doesn't react to it, and we avoid this problem. What I just said was probably a little confusing, so here's a diagram to clear things up. So we have our Rh negative mother pregnant with an Rh positive fetus, and at this point she doesn't produce any Rh antibodies because she hasn't been exposed to the blood of the infant just yet. However, over the course of the childbearing process, the blood will mix and she'll be exposed to the Rh positive blood, such that now she produces anti-Rh antibodies. Then when she's pregnant with her second Rh positive fetus, we develop this hemolytic disease, which is the result of an immunoreaction between the infant's blood and these anti-Rh antibodies. And this disease varies quite a bit, but at its most severe it can cause stillbirths. So this is why we want to give the mother the anti-Rh positive antibody right before right after she's given birth to the first Rh positive infant, so that the infant blood clumps up and is expelled from her body before she can be exposed to it and develop an immunoreaction. And in this case, we don't see any of this hemolytic disease with the second baby because she really hasn't been exposed to any Rh positive blood. So we've effectively prevented any development of a hemolytic disease. Now we're going to talk about platelets or thrombocytes, which are the cell particles responsible for blood clotting. And thrombocytes are not even complete cells themselves, but they're fragments of larger cells called megakaryocytes, which stay in the bone marrow and release these thrombocytes. And these megakaryocytes bud pieces off of their cytoplasm and released what are now thrombocytes into the bloodstream, and they are capable of producing 200 million thrombocytes a day. Or rather, we're capable of producing 200 million thrombocytes a day. And these fragments lack organelles and don't have any molecules to store energy. Instead, they contain packets of physiologically active compounds which are responsible for blood clotting. Once these compounds are released from the thrombocyte into the surrounding plasma, a clot will begin to form, and this clotting process is called hemostasis. So keep in mind, hemostasis is not the same as homeostasis, which is the regulation of all body processes, rather. Hemostasis is just the clotting process. So first, the blood vessel is damaged, which turns the normally smooth interior of the blood vessel rough. And these rough edges catch platelets, and when we get enough platelets clumped together, we form a platelet plug. And in small cuts or wounds, this may seal the wound without any more need for the formation of a true clot. However, if the wound is large enough, a clot will form as the stuck platelets begin to rupture with the pressure of the passing blood, and this allows them to release the compounds they contain into the plasma such that they begin to react with the components of the plasma. And this begins the series of events that leads to the final formation of the clot, which causes blood flow out of the wound to cease. The damaged tissue and trapped platelets release a chemical called prothrombinase activator, which converts the protein and prothrombin in plasma into its active form, thrombin. Now, this thrombin activates the plasma protein, or rather the other plasma protein called fibrinogen, which converts to its active form, fibrin, which are actually long, thin fibers. And these fibers then form a net which catches the erythrocytes or red blood cells, and new fibrinogen interacts with fresh thrombin, and this causes the clotting cascade to continue such that we're still forming a clot. And then when the plasma stops flowing, the clotting has succeeded at stopping the bleeding. So here's a diagram which shows the process of clot formation. And we're not going to worry too much about whether we're talking about the extrinsic or the intrinsic pathway. And we're not really going to talk about the release of calcium. Just know that when the platelets get together, that this eventually leads to the rupturing of those platelets or thrombocytes, such that we release prothrombinase, which then interacts with the protein found in the plasma, prothrombin, and converts it into its active form, thrombin, which then can interact with another plasma protein called fibrinogen, and this converts it into its active form, fibrin, which forms these fibrous threads which acts as a net to catch the erythrocytes so that we stop the bleeding. Now we're going to talk about some disorders of the blood which can be life-threatening or cause acute disabilities. The first one we're going to talk about is infectious mononucleosis, commonly called mono. This is a common blood disease on college campuses, and it's caused by the Epstein-Barr virus, which is actually a virus that can lead to cancer, and we'll talk more about this in the cancer chapter. And this disease is usually transmitted through saliva, so it's called the kissing disease. You can also get mono from drinking after others or putting your mouth on something that someone else's mouth has been on, I suppose. There's no vaccine for this as of now. However, some symptoms of mono can be severe because it can cause your spleen to enlarge, and in extreme cases, this could cause the spleen to rupture and cause you to bleed to death. Another symptom, is just extreme fatigue. Now we're going to talk about a more severe blood disease, leukemia, which is blood cancer. And the symptoms of leukemia are usually flu-like, which means it can be very difficult to diagnose sometimes. And the symptoms include increased risk of infection, swelling of lymph nodes in the spleen, reduction in erythrocyte numbers leading to weakness and fatigue. And another symptom that can lead to a diagnosis of leukemia is bone pain. The actual causes of leukemia are unknown. There are some risk factors like family history, though a lot of times this disease isn't actually genetic. Exposure to certain carcinogens, we're not sure which ones, and also ionizing radiation, and likely viruses also. And we'll talk way more about cancer when we get to that chapter. And leukemia is classified by its pattern of onset and by the specific cells affected. So acute leukemia appears quickly, and in this leukemia, the blood is filled with extremely immature white blood cells called blasts, and these cells pretty much just outcompete the erythrocytes such that the individual is going to have difficulty getting oxygen to the body tissues. In contrast, chronic leukemia occurs more slowly with more developed blood cells. However, seeing as this is leukemia and it's a cancer of the white blood cells, we still have the same sort of problem in both acute and chronic leukemia. And leukemia can be myeloid or lymphoid depending on which type of leukocyte we're seeing affected here. And treatment for leukemia includes chemotherapy, radiation therapy, bone marrow transplants in some cases, and certain kinds of biological therapy. Luckily, this is often successful in leukemia. Anemia is another serious blood condition, and this is a shortage of hemoglobin or red blood cells. And anemia results in a reduction of the blood's oxygen carrying capacity because of our lack of hemoglobin and erythrocytes. And symptoms include fatigue, weakness, shortness of breath, and chest pains. And this is diagnosed via hematocrit, which is a test used to assess the percentage of erythrocytes compared to the total blood volume. So it's the amount of erythrocytes per blood volume. And there are several different kinds of anemia, and these are classified according to the cause of the erythrocyte deficiency. So in iron deficiency anemia, we have a shortage of iron ions, which leads to fewer hemoglobin molecules per erythrocyte. So now we've reduced our oxygen carrying capacity. In pernicious anemia, we have a shortage of vitamin B12, which inhibits the formation of erythrocytes such that we have a reduced number of these cells. In aplastic anemia, the bone marrow simply does not produce enough stem cells to form erythrocytes from. There's also hemorrhagic anemia caused by excessive bleeding, and of course this leads to the reduction of erythrocytes. And finally, there's sickle cell anemia, which is an inherited disorder which creates some mishapen and misshapen erythrocytes which cannot carry hemoglobin as well, if at all, and they have difficulty traveling through capillaries also. And here's a picture of the blood of someone with sickle cell anemia, and as you can see, they have some cells that are pretty much shaped like erythrocytes, but they also have these sickle-shaped cells that have a lower oxygen carrying capacity, and they tend to get lodged in the blood vessel as well. So this is a very serious type of anemia, and all of these different types could lead to death, I suppose. Now we're going to talk about carbon monoxide poisoning, which leads to the prevention of red blood cells from delivering oxygen to your body's tissues. And keep in mind, this is carbon monoxide, not dioxide, so this is a CO. And carbon monoxide is a colorless, odorless gas which can decrease the hemoglobin's ability to carry oxygen. And this is because carbon monoxide binds much more tightly to hemoglobin than oxygen does, and this means that the erythrocytes become saturated with CO such that they can no longer bind to oxygen because they're holding on to CO so tightly. Erythrocytes contaminated with CO float around uselessly through the blood until they wear out or are destroyed because they can't release the CO. And severe CO poisoning can cause starvation, or rather oxygen starvation of the body tissues. And this can cause brain damage, myocardial infarction, or even death. The first symptoms of CO2 poisoning are drowsiness and headache. So one way to prevent CO poisoning is to use CO detectors, which go off when they detect CO, and then move into an area with fresh air if you suspect CO poisoning may occur. And treatment of CO poisoning may require a blood transfusion simply so we can get blood cells that haven't been affected by CO into an individual. There are, of course, many other types of blood disease because pathogens can live in the blood. Many pathogens are capable of traveling within the blood, and these include hepatitis, HIV, and some other sexually transmitted diseases, which we'll talk about in more detail later. Our best defense against bloodborne pathogens is to prevent cross-contamination with someone else's blood, so just avoid contact with another person's blood. Blood banks routinely test their stocks of blood for viral contamination because we obviously don't want to transmit these diseases any more than they have already been transmitted. A good way to prevent any type of bloodborne disease is to avoid unprotected intercourse, as unprotected intercourse can tear mucous membranes, and this would lead to unintentional contact between the two bloodstreams of the two individuals. And also avoid used needles and it's probably best to just completely avoid any sort of recreational intravenous drug use, because these can lead to the transmission of various bloodborne diseases. The topic of the next module is the respiratory system, and I just like to follow the cardiovascular system with the respiratory system because the two are connected and they work together. And the cardiovascular system interacts very closely with the respiratory system to distribute oxygen and remove carbon dioxide. And the respiratory system is responsible for bringing oxygen and air into the body and then removing the carbon dioxide produced as a waste. And it's the topic of the next module, like I said.